Hi plant propagators. In the last unit we looked at some of the basic vocabulary for describing plant parts, and we're now going to build on that knowledge by starting to look at asexual propagation techniques. Vegetative or asexual propagation techniques provide us with many options for reproducing plants, and we'll be looking at the most commonly used techniques in several different modules. In this lecture, I'll be giving you an overview of vegetative propagation techniques that use cuttings and talking about some of the steps we can take to optimize our success when we're managing mother stock and collecting cuttings. By the end of this unit, you'll be able to define vegetative propagation, name the plant parts we can use to propagate plants from cuttings, and describe how to take care of cutting material before it's stuck. Let's start by defining vegetative propagation. Sexual propagation, which involves producing new plants from seed or spores, involves the exchange of genetic material between male and female cells. In contrast, vegetative or asexual propagation involves no exchange of genetic material between male and female cells. The plants resulting from asexual propagation are clones of the parent plant, meaning they're genetically identical to the parent plant. Asexual propagation techniques are essential when we want to reproduce desirable characteristics of the parent plant. For example, perhaps we want to reproduce variegated foliage, as is the case with the two plants pictured here on the right, Coria winds wonder at the top and Diamond Heights ceanothus at the bottom. Perhaps we want to reproduce a specific growth habit. An example might be the Warren a Little selection of California buckwheat, Eriogonum fasciculatum. California buckwheat has an upright growth habit, but Warren a Little has a prostrate growth habit, and we need to propagate it asexually from cuttings in order to ensure that our plants retain the prostrate growth habit. Asexual propagation is also necessary if we're wanting to reproduce specific biochemical properties of the parent plant, for example, a particularly high THC level in cannabis. Vegetative techniques are also essential when we're propagating hybrid plants. If we try to grow hybrids from seed, we'll obtain seedlings that exhibit a range of genetic variability, and there's no guarantee that any of them will be identical to the parent plant. Therefore, we have to propagate the parent plant by asexual methods. Some plants are sterile, meaning they don't produce any seed or don't produce seed that's viable. This means that we have to use asexual propagation techniques for these plants. Some plants bloom very rarely, so again, asexual propagation is usually the only way to propagate these plants on a regular and reliable basis. Another application of asexual propagation techniques is when we want to reproduce predictably a plant of a particular sex. Many plants produce flowers that are bisexual, meaning the flowers contain both male and female reproductive parts. Some plants produce separate male and female flowers on the same plant, and these plants are described as monoecious. Mono means one, and ecious comes from the Greek word for house. So we have two flowers of the opposite sex living in the same house. Some plants produce separate male and female flowers on separate plants, and these plants are described as dioecious, meaning two houses. Sometimes the male or the female plant of a species that's dioecious may be more desirable from an ornamental or a practical or a biochemical point of view. In this case, asexual propagation techniques have to be used in order to reliably reproduce a plant of the desired sex. An example of this is the ginkgo tree, pictured here on the right, which is a popular ornamental tree both in residential gardens and as a street tree. There are actually several ginkgos planted in the new part of Aptos village where all the new construction's going on. Female ginkgos produce fruit and seeds that are messy and smell vile, so nobody wants the female trees. Another example is our California native shrub Garrya elliptica, the coast silk tassel. The male flowers are much longer and a lot showier than the female flowers, and therefore the male plants are much more desirable from an ornamental point of view. There are a variety of vegetative propagation techniques used in commercial nurseries, including divisions and separations, cuttings, micropropagation, layering, and grafting. For the rest of this unit, we're going to focus just on cuttings. A cutting is any part of a plant that's removed from the mother plant, then treated in ways that stimulate regeneration of the missing organs. Depending on the type of cutting, those missing organs may be roots, shoots, or a combination of both. Let's take a quick look at the main plant parts that are used for cuttings. And over the next few weeks, we'll be taking a closer look at all of these different types of cuttings. Stem cuttings are probably the type of cutting that most people are familiar with. Stem cuttings are taken from a section of stem tissue that consists of lateral and sometimes terminal buds. Stem cuttings might be up to 2 feet long or barely 2 millimeters long, and there are several different types. Part of the art and skill of the propagator is selecting the most appropriate method for our particular situation, depending on the structures we have and the equipment to manage the propagation environment, and on the type of plant and species being propagated. Some plants can be propagated from just their leaves, but leaf cuttings aren't used very often because there aren't many species that will produce both new roots and new shoots from just a leaf. Many succulents can be propagated from leaf cuttings though, and the herbaceous perennials Rex begonias, African violet, and Cape primrose are still propagated commercially from leaf cuttings. Other plants that can be propagated from leaf cuttings include some Heuchera species and some Plectranthus. It takes longer though to produce a marketable plant from a leaf cutting, so Heuchera and Plectranthus are usually grown from stem cuttings. And many of the popular Heuchera that are grown for their colored foliage are usually produced by tissue culture. Leaf bud cuttings are very similar to a stem cutting, but they have only one node. Leaf bud cuttings are useful for vines, which often have very long internodes. This technique is also really useful when cutting material is limited, because you can obtain at least twice as many cuttings using leaf buds as you can from stem cuttings, which have a minimum of 2 nodes. The photo here shows leaf bud cuttings of golden pothos, which is a vine native to French Polynesia and is commonly grown as a houseplant in many countries, including the US. Some plants can be propagated from root cuttings. Usually these plants produce sections of root which are thicker and have more carbohydrate reserves. There'll be a unit dedicated just to root cuttings in a few weeks' time. But for the moment, think back to the lecture on nursery hygiene, and in particular to soil-borne plant pathogens such as Pythium and Phytophthora. What do you think might be a problem when we're propagating a plant by root cuttings? If you're thinking that soil-borne pathogens might be present in the soil or substrate around the roots, then you're on the right track. There's a big risk of plant pathogens being spread when we're propagating plants by root cuttings. And we'll come back to that in the lecture on root cuttings. Root cuttings are also very labor intensive, as a plant has to be dug up in order to take the cuttings. So this technique is rarely used in commercial nurseries anymore, but it's still a useful method to have in our toolbox of propagation techniques. As you learned in the unit on basic botanical terminology, meristems are areas of new growth where cells are rapidly dividing. Micropropagation techniques used in tissue culture are able to excise, which means cut out, these tiny areas and propagate them in vitro in a lab under sterile conditions. No matter which plant part we're using as cuttings, and no matter what type of cutting technique we're using, optimizing our chances of success means planning ahead. Firstly, we should make sure the mother plants we're going to take our cuttings from are healthy and not stressed in any obvious way. This means no evidence of water or nutrient stress and no obvious pests or diseases. It's good to try and plan ahead by at least a week so you can fertilize plants if necessary, spray for any pests or diseases, and make sure the mother plants are watered on a regular basis so that they're not drought stressed. Some tree and shrub nurseries maintain stock beds of mother plants that they take cuttings from. Here in California, land is so expensive that most nurseries use as much space as possible for plants that are being grown on for sale. There aren't many nurseries around here that maintain stock beds. Instead, cuttings are taken from plants that are being grown on for sale. This has the added benefit of promoting bushy growth in the plants and therefore producing a better looking plant for sale. Nurseries that are growing trees and shrubs for mail order have to maintain their plants at specific heights so they fit in the mail order boxes. These prunings can be used for cuttings if they're the right size, the right age, and if it's the right season for taking cuttings of that particular plant. Having made sure that the mother plants are well maintained, we need to take good care of our cutting material too. Ideally, plug trays or other containers that are being used should be filled or prepared ahead of time so they're ready for use as soon as the cuttings are brought back to the preparation area. Cuttings should be collected early in the morning while the leaf cells are still turgid and haven't had time to lose too much water through evapotranspiration. Any containers that cuttings are going to be collected in should be surface sterilized before use, and clippers or pruners should also be sprayed frequently with isopropyl alcohol while you're collecting the cutting material. Once cut, the plant material shouldn't be allowed to dry out. Don't leave stems exposed to the sun or the wind. Take a spray bottle of clean water with you and mist the cut plant material lightly to maintain a high level of humidity around leafy material. Large plastic containers with lids are great for collecting cuttings. Ice chests are also good for collecting cuttings. If it's going to be a while before the cuttings are taken back to the nursery, put ice packs in the bottom of the container and cover the ice packs with newspaper or a towel. Don't put cutting material directly on top of the ice or ice packs, as the leaves may freeze. If necessary, covered containers of cut material can be stored overnight in a fridge or a walk-in cooler. Then prepare cuttings somewhere cool in the shade to reduce water loss from evapotranspiration. Don't leave cutting material, whether it's stems or roots, out on the workbench while you take a break. If you have to leave, mist the cuttings with clean water, cover them with damp newspaper or something similar, and make sure they're in the shade. If you're leaving on a lunch break, remember that the sun moves across the sky, or more precisely, the earth moves. So make sure your cuttings will be in the shade for the whole time you're gone, and not just when you leave for your lunch break. Let's finish by summarizing the material covered in this short unit. The terms asexual propagation and vegetative propagation are used interchangeably and are used to describe propagation techniques that don't involve the exchange of genetic material between male and female cells. The plants resulting from asexual propagation are clones of their parent, meaning they're genetically identical to the parent plant. Asexual propagation techniques are essential for reproducing desirable characteristics of the parent plant, for propagating hybrids, sterile plants, and plants which don't flower and produce seed reliably. They're also essential when we want to reproduce a plant of a specific sex. Asexual propagation techniques include divisions and separations, cuttings, micropropagation, layering, and grafting. A cutting is any part of a plant that's removed from the mother plant, then treated in ways that stimulate regeneration of the missing organs. Plant parts that we can use as cuttings include stems, leaf buds, leaves, roots, and meristems. We should always take cuttings from healthy mother plants that don't show any signs of stress or pests and diseases. Once removed from the mother plant, cutting material should always be treated in ways that minimize water loss through evapotranspiration in order to increase the chance of the cuttings producing roots and, where applicable, shoots. That's the end of this short unit. Next, complete the short quiz to test your understanding of this material.